time she's going to be here?
She's coming Thursday.
Okay, so anytime Fridays up.
Somebody started recording. Let me go to a different place. Linking soil biological health to essential functions in the southern highlands. Any interest?
Not right now.
Her name is Veronica Acosta Martinez.
Dan Devlin, you might want to mute your line.
Yes.
Somebody was asking me who was speaking this morning, and I told them, and they didn't know him. And I said, I think I do.
Yes, you do.
Hi.
How are you doing, Amy?
Good, thanks, everybody. So we'll just wait maybe another minute or two before getting started. Just for those of you who weren't here a minute or two ago, just so you know, we are recording this session. If you could mute your lines so that there's a minimum of background noise, that would be excellent. And please feel free to use the chat box. If you hover over your screen, you can raise the chat box and you can post your questions there that we can field, and basically I'll read them out at the end of the session and we'll do them one at a time in case anyone calls in. That way they can hear the questions and we don't have to worry about muting and unmuting you. I'll just read them out. Maybe we'll wait one more minute, okay? Is that all right? Okay, I can continue to let people in as they come, but how do you feel about getting started now? Is this good?
Okay.
All right, wonderful. Well, welcome everyone, and thank you so much for taking time out of your busy schedules to join us today for this webinar. It's my great pleasure to be able to be the host today. My name is Amy Kremen. I'm the project manager for the Ogallala Water Coordinated Agriculture Project, which is a 4-year project that's been in operation since 2016 that's funded by USDA NIFA. We are a large group of researchers, students, postdocs with an advisory board of farmers and others that has been collaborating for the last 4 years, since 2016, on a range of integrated research topics spanning dryland to fully irrigated systems. And we are delighted today to be able to focus, shine a spotlight on some members of our team who have been researching soil health in the Southern High Plains, Drs. Veronica Acosta Martinez and Rajan Ghamiri. So we just as one final note, we are recording this session, and we encourage you to put your questions into the chat box, and we'll field them at the end of the call. And if you could please mute your lines for minimizing background noise, that would be most appreciated. And so with that, I will turn things over to Veronica and Rajan. Thank you so much for presenting today.
Oh, good afternoon, everyone. Thanks to Amy, Megan, and the organizers of this webinar series for giving Rajan and I an opportunity to discuss about the soil health and our challenges in the Southern High Plains. And thanks to all the participants today for joining us. And when we talk about soil quality/health, we're referring to the capacity of the soil to function within its ecosystem boundaries and the chemical, physical, and biological component. With recent soil health initiatives, soil conservation efforts, maybe beginning since 2015 in the International Year of Soils, we are trying to focus in the soil health in terms of the looking and highlighting the biological component, giving also importance, listening to the biological component and the soil as a living system. We're trying to support a diverse and active biological community, as you see there on the circle on the left, that will support, provides essential functions for all of us. The soil health team of the Ogallala Aquifer Project represents different locations of the Ogallala Aquifer region and different expertise and indicators, the specific methods that we're using for our soil health assessment across this region. That way, we're trying to synchronize our resources and compare soil health using the same indicators and methods. You can see there the familiar faces of our team, and I just wanted to explain that in the Southern High Plains region, Chuck West and I have shared different postdocs, like Amanda Cano at the beginning of the program, that help us to develop a soil health review that I will be discussing soon, and that is part of an effort with all our team. And also we have a soil health fact sheet that can be found in the Ogallala Aquifer website. And also we'll have here on the picture with a white hat, Krishna Bandari, that have joined also our team and is helping us also. In my team, I have Lumari Perez Guzman and John Cotton. And Rajan also will be talking about the different projects and different students that are involved. So establishing indexes of soil health across a region requires to understand the complex interactions of the management, the soil properties, the crops, and the environment. And the aquifer region offers a variety of management, crops, environmental interactions, and inherent wide range in inherent soil properties. So we need to look at sensitive indicators like the soil biology is. And in this case, then when we look at the top three, the bacteria, fungi, and protozoa, they are the microscopic, they are therefore also then higher in numbers and in diversity. And they are the microbial component of the soils, which is linked to essential soil functions. They are involved in 80 to 90% of the decomposition processes that occur in soils and accounting for soil respiration mainly, generally. And also they are involved in making nutrients available in our soils, carbon, nitrogen, phosphorus, sulfur. So they regulate the biochemical potential of our soils. They also influence the soil organic matter and therefore brings a wide variety of benefits with the organic matter dynamics and biodiversity that it can bring. It also then helps with that way with the biodegradation, bioremediation potential, detoxification capacity of our soils, and also can bring symbiotic and compensatory associations that are essential for our soils. An example is the rhizobia, bacteria plant associations that help plants fix nitrogen, and the AMF, arbuscular mycorrhiza fungi association that helps plant also to explore environments it wouldn't be able to without these fungi, and helps also the plant in obtaining better water and nutrients. And it also is important in carbon dynamics. So the changes in soil organic matter will lead to better aggregation of our soils, improve the soil structure, and also help in increasing water holding capacity of our soils. So it can bring benefits to the soil water storage and erosion control potential of our soils. And recognizing the importance of microbes to the soil health and functions, we need then rapid, inexpensive, and sensitive methods to evaluate them. So in terms of measuring the soil microbial component, I have tried to do that and to use the same methods over the decades that I have been in this region. I use the chloroform fumigation extraction method, in which when you kill microbes, it releases carbon and nitrogen from the microbial biomass that can be then measured. And provide indications of the microbial communities in terms of size. You also can extract fatty acids from the microbial community from our soils. More than 100 fatty acids can be extracted, and their abundance can be added, and that total fatty acids can give information also of the community size. In addition, looking more at their fluctuations in the fatty acid profiling, you can gain information on who's there, the microbial community composition in terms of the fluctuations of fungal versus bacterial populations. As there are also specific markers that I have looked through the literature over the years and have in libraries to compare with given markers, the saprophytic fungi changes that are so important for the composition processes. There is one marker available for the AMF, the arbuscular mycorrhiza fungi, that I describe is one of the more beneficial microorganisms that you could also evaluate and find their changes over time. Also, use markers for gram-positive bacteria that provide inherent resistance and inflammation strategies to cope with stress to the community. They also use certain type of carbon sources that differ from gram-negative bacteria plus other functions that they perform. And to the gram-negative bacteria with these markers, and there are others also that I have been using, you can obtain information of protobacteria that are important in carbon cycling. Also rhizobia are members here, this group. And there are markers for actinobacteria that are like counterparts of fungal component in being also good in the case of bacteria that can decompose complex substrates in soil. So looking at fungal and bacterial ratios can provide also some information of a beneficial shift in soil health. We see how they different roles. Increases in fungi can provide information. It can provide signs of benefits to aggregate formation, improvement in soil organic matter dynamics, that they have better carbon utilization efficiency. You can also use many other markers, ratios, and indexes. I could suggest also the gram-positive and gram-negative bacterial ratio, as I was describing how gram-positive use more recalcitrant carbon sources than gram-negative bacteria that rely so much in the labile. So you could obtain with these ratios even over time and changes on the management that you're applying. Look at how also soil organic carbon dynamics is being modified. And overall, also, you could look at the diversity more in terms of the changes with DNA profiling to the species level. It's just that this data is a little bit more intense. You obtain more data than with other techniques. It takes just more time and effort to look at this data. So I use also this type of methods with certain projects. But overall, the changes in the size and in the composition of the microbial communities influence many functions. We evaluate the biochemical potential of our soil, evaluate the enzyme activities. So assessments of different enzymes involving different reactions in soils for carbon, nitrogen, phosphorus, and sulfur cycling have been valuable to obtain information of the biochemical potential of soils. Also, then in our team, we have that Rice, Rajan, and Megan do more research on the functions related to organic matter dynamics, as they can use different carbon pools to evaluate the changes in the organic matter in terms of chemistry over time. And there are certain pools that are more active and labile and can give indications earlier of the potential changes in improvement in carbon sequestration of the soils and accumulation. So the Southern High Plains region that is shown in this map here in this rectangle covers the eastern New Mexico to northwestern Texas. And in this region, what this map is showing from the climate.gov website is how in this region we have the highest withdrawal rates on the Ogallala Aquifer. So we experience the largest declines as shown in the brown areas in the region. More than 150 feet of water level changes can be experienced. These are data from recent years. And these are challenges for the region, and it impacts the agriculture. So far, this is one of the major cotton producing regions of the United States. More than 1.5 million hectares are under cotton production, and with 50% being irrigated from this aquifer, but this percent is going down. That's a fact. But it has been also important in sorghum production, and mostly also due to the transitions that we're experiencing as it is a more drought-tolerant crop. So within cotton, sorghum, the corn production, the byproducts of the cotton ginning supports a large beef feeding industry as well, and a smaller but significant dairy industry. And this region, I will say that could be challenging to sustain soil health, but it's possible. And what this map is showing is how, in the rectangle also, the Southern High Plains region is a region that has the highest wind erosion rates per acre. A lot of tons, more than 15 tons of dust can be produced per acre. It's one of the most persistent dust producing regions of North America. And this is due to different factors, the flatness of the land, the high wind speeds, more than 5 meters per second. And these soils are generally porous, low organic matter content, and are exposed to dust storms, events like this that resemble the conditions of the 1930s. So in order to look for changes and improvement in our soils, our management will matter. Generally, also these soils are exposed to hot temperatures, several consecutive days, more than 90 degrees during the summers, and low precipitation, less than 20 inches of rain is received in this region. And climate is becoming more variable with more extreme and record droughts. So this is a challenge on how the soils are exposed to these conditions. With the management, when we have more monocultures, then it would involve more winter fallow periods that exposes them the soils more and make it them more prone to large-scale wind erosion. So in terms of the transition to dryland in the Ogallala Aquifer, we are experiencing this transition in different ways in this region. Here for us, it's going at a faster rate. But overall, we're going to face going from the potential with irrigation and more water available to have more soil moisture, more biomass production, and be able to accumulate more soil organic carbon, to the extreme in dryland with climatic variabilities and more frequent droughts to have more fallow periods, and then reduce the biomass that is produced due to lower moisture, and the potential to accumulate soil organic carbon. So the soil health will become more and more important. How we could improve the capacity of our soils to hold more water from precipitation, which is free, and is what we will have. And what we can do here is we can control our management selections as possible. If there is amendments available and it's possible to use them, they will help. Cover crops, crop rotations that Rajan will explain today, the conservation tillage associated to these. In Texas, we're having a recent transition in many enthusiastic efforts towards no tillage practices. I work with a producer, Lacey Vardeman, that have more than 11,000 acres that they're farming, her family. And there are many different scenarios here of different years in which they have tried the no tillage, and I have even compared within the first year of the transition to no tillage and see improvements in the microbial component. And also then livestock integration has been an option to reduce the water needed in the systems. And Vivian Allen have done research for the past decades in Old World Blue stem for this region to integrate with the maintaining cotton production, but systems that have improved the microbial biomass carbon and some activities of our soils, the biochemical potential, but also reduce the water needed and the fertilizers. And West have done research with me evaluating the introduction of alfalfa and the effects on the microbial component. And that integration have further also improved the microbial component and reduce the fertilizers needed into none, or also the water needed as is possible in dryland. And the CRP has been also an important conservation effort in this region. Texas leads the nation in the conservation research program. And recently, then more contracts then could expire. So it's important what we do. Within the first research with Jennifer Moore-Kucera and Cheng-Wei Lee and others, we have shown that the microbial component can be improved within the first 13 to 15 years of this program. So also to look at what will happen when we take out this land after this soil biological restoration is happening. With John Cotton, we found out that within the first month of converting CRP to row crop and the 4 tillage passages that were done, the microbial biomass carbon was reduced by 50%. So organic carbon by 33%, and different enzyme activities, especially beta-glucosidase, also a soil health indicator in carbon cycling, was reduced with a phosphodiesterase activity by 60 to 70%. So what we do when we convert also land like this into agricultural practices matter. Things that we cannot control are the soil types, the climate variability, and the Ogallala aquifer water available. And I wanted to show this view note from NRCS because it is very interesting and encouraging. And we could think about it and discuss it also later, in which they were trying to do a relationship that every % of increase in organic matter could result in an increase of 25,000 gallons of water available per acre. So this could be challenging. It depends on the region, on the soil types, and the management combinations. Maybe 1% of organic matter is not that easy to accomplish in few years. But to keep this in mind, to have this information available, and think also that the recent studies have provided direct evidence that the soil microbes are responsible for the formation of chemically diverse and stable soil organic matter, so that they are important even if you cannot change in percent that on the organic matter chemistry and stability that will lead to these improvements in water, salt water holding potential, so that we keep in mind that they could be signs and indicators that we could use of early changes that this can happen. So the transition to dryland production in Texas is happening faster than other regions. As I was describing the transition from irrigated to dryland, we have evaluated different alternative management to save water with the practices, and how we have seen that it can also help and improve the microbial component. Something that we also wanted to do is to find research related to the irrigation strategies. And we were able to find in 2018 producers that were assisted to change from center pivot irrigation to subsurface drip irrigation some of their land. And we were able then to take soil samples after 1 year of this happening and 2 years of this change. And this was a program with NRCS assisting them to do this. So we were able that way to take soil samples after 1 year of the change of this water usage in their land from the center pivot irrigation, and then from the subsurface drip. And some of them were able to have other crops, mainly cotton production. And also from the dryland, we took soil samples depending on the management. Some of them were able to have wheat. And also we found a CRP land that was established in 1986. So for a long time, I wanted just to show you how even visually you could see an Alton soil series, sandy soil, how it can, for so long, almost 30 something years, can get to look more aggregated over this long soil restoration process. And it also have 3 times greater total fatty acids, an indication of microbial community size, compared to the nearby land under this management. And also then it has 10 times higher AMF compared to the row crop nearby. And I can't see my presentation. Something happened. And in this case, then we have a high coverage also with these three producers of about 476 hectares of land and about 13 different sites and scenarios that we're evaluating in this region of sandy soils with these changes. And I wanted just to give an overview of the changes in management and how it has impacted the microbial community so far with 2 years of this transition from irrigated to dryland. So that would be my next slide.
I'm not sure. Let's see. Hold on. 1 second. 1 second. Rajan, just checking with you. Looks like something happened to interrupt our meeting.
Okay.
No problem.
Yeah, looks like
It's okay.
now it is. I think maybe something is changed.
Amy, I think we were on slide 17.
Okay, good. Thank you.
Wonderful. Thanks, Susie.
Yeah, no problem.
Yeah, it's interesting. This is the first time we've used Teams for a webinar. I didn't know it could be interrupted, but that's okay. We'll get there. Thanks, Rajan.
We are on this slide.
Yeah. Oh, keep going. Let's see. And I'll take control. So doing this evaluation within different sites that offer this variety of changes from center pivots of surface drip, dryland. What I wanted is just to give an overview first. I will not present too much data yet, as I want to evaluate things over more time. But the microbial community composition is showing to differ in the transition from irrigated to dryland after 2 years only. So here you see a principal component analysis in which what we did is that you can evaluate the soil samples with all the fatty acids you extract per sample. So here I'm showing that per field, per system, we took 3 soil samples in a field, and then that way, the middle points that are close symbols have an average per system. So in that case, evaluating so many fatty acids, the fatty acid profiling representing the microbial community composition is showing how the composition of the communities in the center pivot differs. Is in one quadrant compared to the community, microbial community composition of the subsurface drip irrigation systems. So the water matters. Also, we try to evaluate generally vectors of those markers, microbial groups that I described before, the AMF, fungi, and bacteria, but they're not pointing in a particular system to be higher in a particular system than the other yet in this comparison. We also here are trying to compare another a group of systems from another producer, and it showed also that another subsurface drip irrigation will have a different microbial community composition compared to the nearby dryland. And in this case, when we look more at these vectors, they are pointing to the system that have this dryland having higher AMF, fungi, and bacteria, possibly due to having a winter cover crop rye in the management history. So it shows that even being able to add a winter cover crop in the management history could then maybe compensate for having then being under dryland rather than being with some irrigation under a given irrigation system. So overall, this transition is happening and is showing that the microbial community's composition differ because of a decrease in the microbial community size as we go from center pivot to the subsurface drip. There are decreases in the microbial community size, the microbial biomass carbon. You keep seeing the decreases in the community until you go to the dryland system that has generally then the center pivot double microbial community size than in the dryland. But I always wanted to emphasize how in this case, even this early, we can see that this system, the dryland, possibly having that rye cover crop, have twice double the microbial community size than the subsurface drip system. So Within this transition, then it's possible then that what the producers are able to do is they're able to use other management conservative practices, amendments, or winter cover crops. It could help with these reductions that will be experienced in the microbial communities from the change from center pivot to subsurface strip to dryland. And this is something that then we will continue doing soil samplings and look at this management history. And Rajan will talk about research plots that has been established for longer. We could look at the future and the evaluation of other options of cover crops that then we could learn from their benefits.
Okay. Thank you, Veronica, for a nice overview of soil health in Ogallala Aquifer region. Challenges we face in this area, and also various management options that could improve soil health while transitioning from irrigated to dryland production. In my presentation, I will be specifically focusing on a cover crop study we did in eastern New Mexico, and then relate that how cover crop can improve soil health and provide other benefits, to the producers who are changing from irrigated to dryland situation. There are various different kind of crops used as a cover crop. For example, legumes are used specifically focusing on fixing nitrogen. They can increase nutrient availability in the system. There are grass type cover crops also can be used. Specifically grass type cover crops are good in biomass production. They make good biomass and then provide good ground cover. That's why they are good for controlling erosion and also weed control. And high biomass input also relates to high carbon storage in the system. And also, brassica cover crops are commonly used. Those brassica cover crops are specifically good for reducing compaction in soil. And also, they grow very fast and then take a lot of nutrients. So they are good nutrient scavenger. Then later, they can recycle that nutrient back into the succeeding crop in crop rotation. Looks like someone's window is coming again. Just a second, let me upload the presentation again.
That is very strange.
Yeah, I think, I don't know what is happening. But anyway, these different, when it is loading, these different cover crops provide different functions. Then that ultimately helps in improving soil health.
Mm-hmm.
Let me do it real quick.
Just pause for a moment.
Oh. Okay, yeah, these different cover crops provide different functions in ecosystems. So we designed a study, to demonstrate how these different cover crops help in improving soil health, and then what are their the relationship. And we established a study in 2015 at Agricultural Science Center, Clovis, New Mexico, with 8 different cover crop treatments. Those treatments include fallow, pea, oat, canola, pea and oat mixer, pea canola mixer, pea oat canola, and then more diverse mixer or 6 species mixer of pea, oat, canola, barley, hairy maize, and forage radish. These treatments were designed in a way that fits into winter wheat sorghum fallow 3-year rotation. This winter wheat sorghum fallow 3-year rotation is a typical for dryland or very limited irrigated condition. And then that rotation leaves long fallow period after wheat and sorghum. So our cover crop study was targeted to reduce that fallow period. and use cover crop on instead. So since the region is already limited in irrigation, we designed the rotation in a way that cover crops do not get any irrigation, but winter wheat and sorghum gets a little bit of irrigation. For example, in typical irrigated scenario, winter wheat needs about 20-inch irrigation on this area, and sorghum needs 10 to 15-inch irrigation. But we are reducing that at least 50% or less than 50% of irrigation need for those main crop. And again, cover crops, we did not provide any irrigation to grow them. Then when we look at changes in soil with this cover cropping practice, we could see a lot of increased root activity, increased nodulation, specifically on legume plots. You can see in this middle picture that the rhizobium activity happening, and then we also see progression in earthworm activity, increasing number of earthworm. These are our observations in the field, but we also monitor different soil health indicators. Here I'm going to discuss microbial community composition. In collaboration with Veronica, we did collect sample from here and then analyzed those different fatty acid markers on her lab. And then this gives, these different dots indicates different microbial community composition. For example, if you look at this red dot, that indicates fallow treatment. And then all the other dots are moving away from this fallow plot. So that means the community in cover crop system is different than in fallow system. And if we look at these two cover crop treatments, oat and 6 species mixer, these two cover crop treatments move far away from this fallow treatment. And then we find that oat had highest biomass production as compared to other cover crop treatments. And 6 species mixer, of course, it is most diverse. So that is interesting, having more biomass or also having more species mixer, that is helping to diversify, below ground microbial community as well. And we look deep into like what may be the driver of this change in microbial community, and we could see, major change in fungal component. Now you can see all cover crop treatments have more fungal mass as compared to fallow treatment. And if you look at these two treatments, oat and 6 species mixer, those have the highest fungal biomass, both saprophytic fungus and mycorrhizal fungus. So that was very interesting. Fungus is driving changes in microbial community in cover crop system. And we look at not only fungal and bacterial community, and other indicators, other parameters that affect cover cropping system. For example, all fungal component, all bacterial component, and most of soil organic matter components show positive response with cover cropping. There is a positive change. That means increase in those indicators. But if you look at these, couple lines in blue color, total nitrogen, labile organic nitrogen, inorganic nitrogen, and then the other indicator, not actually the soil health, but soil water content. Those there we could see some negative trend, or that means maybe the part of the nitrogen in the system is utilized by cover crop for their growth, and then also water is utilized by cover crop. So this is very critical. We talk this hot environment, dry situation, and then low fertility soil. So we should be looking into benefit versus trade-off on this system. There is a possibility of improving soil health using cover crops, but we need to be considerate of soil nitrogen and also soil water. So on that decision making, we need to look at selection of cover crop species, which use minimum water and then provide maximum benefit. And then also planting and termination timing, which is optimum for your specific farming situation. So that is also important. And water used by various cover crop species and their combinations, that is also critical, and nutrient use and recycling. If we look at the different species of cover crop we compared on our study, it looks like a brassica takes up nutrient very fast, but also they can recycle that very quickly. They decompose faster than legume cover crops. And but when it comes to farmers' decision making, maybe the economic viability of overall system is more important than anything. And that is critical for them. So we work with economists at New Mexico State University to look at different scenarios. What is the net return from cover crop system versus winter fallow system? If we just do simple cost benefit analysis of cover cropping versus living land fallow, the cover cropping systems is a little more expensive, and then reduce net return. But we just discussed that this cover cropping system improves soil carbon, increase nitrogen in a long term, and then also another benefit could be gained from integrating livestock into cover crop system. So we look at the scenario, increasing soil carbon, increasing carbon, and then grazing, and also nitrogen benefit from those legume cover crops. In that situation, if we have more than one component, we can see net benefit from cover cropping as compared to fallow. So this is very critical, consideration, because we need to look into all aspects of cover cropping when we make a cover cropping decision. So among these, various factors I just discussed, I wanted to look at how livestock integration could improve soil health and then support this cover cropping rotation. If you look at eastern New Mexico, West Texas, our entire southern high plains region, livestock is integrated already in our cropping system, either in the form of forage production and use, or in terms of grazing in a standing crop and cover crop residue, and grazing those cattle in native pasture rest of the year. So it is already part of our system. So it would be very nice, to have our cover crop study and then also integrate grazing. But because of our logistic challenge at Egg Science Center Clovis, we could not integrate the grazing component in our study. But I work with neighboring farmers around here to look at how livestock, specifically cattle, is playing role in soil health in this farming systems. So we look at two systems at Egg Science Center Clovis: no-tillage cropping system, strip-tillage cropping systems with the 2-year rotation for last 4 years. But these two systems were in winter wheat sorghum fallow rotation for a long time. But we did not have livestock in the facility or in these plots. They were never grazed. But we had adjacent farm field which was in winter wheat sorghum fallow rotation, but with livestock grazing. So that is a good opportunity to compare these different, grazing versus non-grazing system. And also next to those grazed system, we had 2 grassland. One is with grazing, another without grazing. And then they were in those practice for a long time. So that is, also we included on that comparison. And when we look at different indicators of soil health, for example, looking at biogeochemical cycling, we can look at different enzymes, and we evaluated activity of different enzymes on these systems. Here, I'm just presenting data from beta-glucosaminidase enzyme, because that is related to this organic matter cycling, carbon and nitrogen cycling in agroecosystems. And I think you are seeing this blue bar and then red bars. those blue bars indicate livestock integrated system, and red bars indicate no livestock integration. In case of grassland, in beta-glucosaminidase activity, we don't see increase in beta-glucosaminidase, this enzyme activity with livestock grazing. But in cropland, there is a clear difference. There is an increase in that enzyme activity with livestock integration. And we also look at different organic matter components. And then this potentially mineralizable carbon is one of the indices that microbial activity happening. This is the very labile pool soil organic matter. It changes rapidly. Then we can look at how the system is responding to this change. Again, those blue bars, in this case, in grassland, you can see significantly greater potentially mineralizable nitrogen with livestock integration than without livestock. In case of cropland, if we would not have livestock, the typical scenario is conventional system would have less potentially mineralizable carbon than no-tillage or strip tillage. But in our study, we are seeing this livestock integrated cropland system or conventional till system is almost comparable with no-tillage system. So that is also positive change because of livestock, having livestock in crop rotation. And we look at total soil organic carbon in a 3 feet soil profile. And when we look at that, the total organic carbon in the profile in a grassland, although it was grazed, it is same amount of soil carbon as ungrazed system. And in cropland, it is in fact this grazing system, those blue bars have highest amount of carbon than no tillage and strip tillage. So we can say, having livestock sometimes is as effective as having some other conservation practice like no tillage, reduced tillage, or having permanent perennial grasses. And on this study also, we look at microbial community, again, using those different indicators, different fatty acid profile. And we can see clear separation of grassland with cropland. This GGL and UGL, those are grassland, then others are cropland system. So that difference is more clear than any other thing. But you can also see difference because of livestock grazing. For example, this CTGC and then GGL, these two systems, grazed systems, are more closer than other systems. So that is also interesting. And we look at maybe what is the driver of this change. And in this system, we also see change in fungal community like cover crop systems, but also we could see greater change in gram-positive bacteria and actinobacteria. That was also interesting finding that it can change those community as well. So with that is we are seeing a lot different changes in different components. You are muted.
So, Mary, we have in this region of southern Philippines, we find more challenges to improve the soil health with low fertility soils, hot and dry environments as we have. It could be more challenging than other regions. However, I wanted to point out how the alternative management for this region that have been adopted over time due to the water limitations and changes that we're having have shown that it has potential to stimulate the microbial community and improve soil health and the sustainability of the region. So understanding the role of soil biology and their link and provide that link to how they can help with essential soil functions will help us in keep designing best management practices. And one of the examples is then the research, the long-term adoption of perennial grasses and cattle grazing that have shown that can have benefits in improving soil health and the biochemical cycling and soil organic matter dynamics, and also help in reducing water need, fertilizer needed, and other ecological benefits also on biodiversity.
Cover cropping is also beneficial. We see a positive change in soil organic matter components and microbial community because of cover cropping. And to summarize overall our findings from the studies we just presented, and then our work as a soil health team, is specifically diversifying cropping system, reducing soil disturbance, and aiding organic matter in any form, or integrating livestock and cropping system is beneficial despite hot, dry environment and then low fertility soil. And then we are rapidly changing from irrigated to dryland system. That we need to always consider those things when looking into these different options. And as we have been talking, as a soil health team, we are working a lot in different aspects of soil health management. And more information about our work as a soil health team is available in this Ogallala CAP website. Like we mentioned earlier, we have a fact sheet, and then there is a link for different publications coming from different states. So you can go ahead and check more resources. With that, I would like to thank everyone for coming today. And then I would like to thank collaborators, graduate students involved on the project, postdocs, and then collaborating producers, you who is instrumental on this effort, and all the soil health team and all helping hands. With that, we are ready to take some questions if you may have.
Wonderful. Thank you so much, Rajan and Veronica. We do have a couple of questions that I'll read out because we do have some people on the phone. The first question is there a reason that oats were used rather than wheat? Thinking this is as a cover crop.
Yeah, we used oat because one is winter wheat is already on crop rotation. We did not want it to use same crop, but also oat. If we put oat, they can make faster biomass. If we put on a spring, they can make big biomass faster. And if you do in winter situation, winter cover cropping situation, those are winter kill. So you can put oat in August or September, and then by December, because of the freezing, you can get winter kill. So because of those few benefits of having oat, we pick oat as a cover crop than wheat.
Are there other cover crops that are being studied by your team in New Mexico now that weren't part of these studies that you find interesting?
Beside this study, we did 2 years work in dryland also with pea and winter rye as a cover crop, on the mixer, and then in that corn sorghum rotation. That is one study, and we are also looking at different combination of cover crop. Based on first 3 years data from this study, we see that mixer is better in some of the indicators. So we designed another mix with triticale, hairy beds, and pea, clover, all other different kind of mix, and then look at their response in forage corn and sorghum system. So the study we discussed today is a grain crop rotation, grain sorghum and wheat. And then we have another study, different cover crop treatments, and corn and sorghum in rotation. And then there, again, cover crops, we did not provide irrigation, but forage corn and sorghum, we provide a little more irrigation than this study. And then there, more interesting is when we terminate cover crop, we can see similar kind of negative moisture, like moisture is decreased. But when we look at corn and sorghum yield, there we could see significant increase in corn and sorghum yield when you have a little more water for the main crop. So that is interesting on other study. But that is only like we are on second year on that study. So I am excited to see how that goes over next at least another 2 years. We can look at it.
And just one quick note: when you say you're using just a little bit of irrigation on the main crops, whether for this study that you presented today with the grain crops or for the forage studies, can you just define how many inches of roughly, what's the range? Because you're using very little water, but when you say that, how much is very little?
In this winter wheat sorghum fallow, which I presented today, this one maximum 10 inch. If there is a good rain, and then we feel crop is not, desperately needing irrigation. Some years we give just 7 inch, some years 8 inch. And then in sorghum, looks like this year we just gave one irrigation, that is 1.5 inch. And then we are getting rain, so we are, stopping right there. So 5 inch is maximum on dry year. For example, 2017 was a very dry year on this area, and then there we needed to give at least like 3 irrigations. That made it 5 inch. But we try to give just like we call life-saving irrigation for the main crop, because we are limited. We cannot give, even if we say like we give full irrigation, we cannot give like what it needs on the right time. In forage, our maximum cap is 12 to 15 inch in that forage rotation. And because of the corn, that needs more water than wheat or sorghum. So we are giving up to 15 inch on that rotation.
A very quick follow-up about the oats. Can rye be planted very late in the season?
We used, winter annual rye. On that experiment, we planted in November here in Clovis. So that was successful. We got some successful establishment. Under dryland condition, I would not say that is best. That is excellent, but provided the moisture in the soil, it was a good ground cover for dryland.
So Chuck West, I see you have your hand raised. I will call on you in just a second. We're just going to take another question or two first, if you don't mind. So we have a question from Mark Marsalis. He is asking, how did you put, this is another question for you, Rajan, how did you put a dollar amount on soil carbon in the economic figuring, such as, the net return plus the soil carbon? How are you able to, put a dollar amount on the soil carbon?
Yeah, there are different metrics people use. For example, when they talk about carbon budgeting, they use different metrics. And then we did not have that matrix for New Mexico. So we use some of the literature value. I need to go on the paper, like what exact number we use. But for example, California carbon credit, they give some dollar value. For example, you increase this much carbon, you can get this much credit. So we use that as a basis. And I think I need to follow up with economists, what we use. We discussed a couple options, but we used already standard published data to quantify if we increase this many tons of carbon or this percent of carbon change. And there is a value available for erosion control as well. For example, I remember erosion control of 1 ton is equivalent to $15 or something like that. Mm-hmm. So they provide those dollar numbers, and we use that. And in case of grazing, we use our biomass production and those different cover crop treatments, and then standard, market value again for that also. So that was the basis in our study.
Excellent. Thank you. So, Veronica, here's a question for you. What are your thoughts regarding the use of PLFA for quantifying arbuscular mycorrhizal fungi?
Well, I think I could tell people also to watch for a paper that was just accepted from our team in the Soil Science Society of America journal that will come within few days available, that in which we compare that technique with the one that I use more, the ester-linked fatty acid methyl ester profile, the ELFAME, because generally with the ELFAME you can rely on using it for that marker for AMF. You're not extracting the phospholipids first, you're working first and doing in situ reactions right there of all fatty acids. So you're dealing with all fatty acids coming from different lipids in the soil, and you could then know that you're dealing with fatty acids like the AMF, and that you're working with that one and able to quantify that one. But with the PLFA technique, you're having to get rid of certain type of lipids phases, and you don't include one that will have the AMF spores and this type of AMF. So it's not as reliable to be used for this marker. So there is a lot of controversy there on using it. So I advise always to check. Even there is a company, the MIDI company in Newark, Delaware, that is trying to modify that technique, PLFA, to include the neutral lipids, and then being able to use that technique, the PLFA, for the AMF marker. These things are discussed in this paper in more detail for soil biologists. And it's important that if you also go to a commercial lab, you maybe check what is the technique they're using. I'm always available. I get emails often with questions like this, and I will be happy to help people, producers that would like to send samples to a commercial lab and maybe evaluate for these type of markers, or contact me if they want to evaluate some of their samples.
Wonderful. Chuck West, I made you a presenter, but I'm having a hard time unmuting you. Are you able to unmute to ask a question or share a comment?
Yeah. Yes. Can you hear me okay now?
Yes. Thanks.
Okay. Yeah. My question, actually, this is a point that something I ran across quite unexpectedly a few years ago. I was talking to a producer, actually someone who grew up on a farm in West Central Oklahoma, about using rye as a cover crop. And he said, "Oh, no, not rye. Rye is a weed." And I said, "What?" "Well, yeah, out here in wheat country, we don't want rye out there, because if we get contaminant, that is, if some of that would fall on the ground and germinate in the fall when we plant our wheat, we'll have rye contamination next year when we harvest the wheat, and then we get docked." And so I was very surprised at that. I never heard that before. But that is something you might run across sometimes in the wheat growing areas. That I said, yes, but if you manage a cover crop correctly, you won't have mature rye seeds if it gets grazed or whatever. But there's some escapes that could produce seed. But just to make a point that was a concern in that the wheat producing area of Oklahoma.
Okay, thank you, Chuck, for bringing that. That is, I also get that question or that comment. We don't have in Southern High Plains, we don't have that luxury. We can wait that long. Rye or barley, oat, this can make seed. Most of our cover crop treatments we establish, and then first flowering, whatever the species mixer, when they go on 50% flowering, we terminate those. In most of our experiment, that is the best way we can balance, that water I was talking, water issue, but produce good enough biomass. And also, if you have if you terminate those cover crops around that flowering time, that decompose, and then the succeeding crop can get nutrients from cover cropping that we have seen. Now, for example, on this study, we did one decomposition study, how fast these 8 different cover crops we used, they decompose. And then on that one, we terminated cover crop on flowering stage, and then use that residue. Looks like brassica or that canola decomposed very fast. release nutrients before winter or during winter. And then pea is kind of in middle. But oat also starts releasing some of the nutrients, if you go on February, March next year. So that is, looking into those closer observation, when to terminate, and then how that nutrient is recycled back is also important. Yes, that is an issue. Then if you are late, even though it doesn't make seed, that rye regrows. That is also tough to kill, if it is too long, if you leave that too long.
Fantastic. We'll do one more question because we've just passed the hour and want to be mindful of people's time. This is a question from Megan. Rajan, you mentioned the potential advantages of brassicas in retaining and rapidly recycling nutrients. Have you seen similar effects with the integration of canola into rotations, or do you expect that you might see something similar in diversifying rotations to include brassicas?
Integrating canola also has that effect. It makes good biomass and also recycles nutrient. But if you put canola early, we have seen situation, for example, if you put canola early, grows too much biomass, but that also uses a lot of moisture. That is a trade-off there. It grows good, recycles back fast, but also we need to be considerate on moisture use. And then in another study, we use turnip and radish. There, I feel half of the turnip and radish got winter kill, especially turnip. And then, that is good for reducing compaction. And, the turnip came back in the spring, maybe half of it came back. Then that was also good. So there are different, brassica have different properties. So maybe a mixer. I have not done, different kind of brassica mix. But we need to select the right species. What is your goal?
That's excellent. So, thank you so much. Oh, let's see. Let's just stretch to the bottom. We've got, yes, so as Dan just put in the chat box, thank you both for a very excellent and useful presentation. We are grateful to you both for the time that you've put into putting this together and for your engagement on these topics with our team throughout the project. And we're also thankful to all of you today who joined us today and engaged and added your questions. We will leave this chat box open for the next hour, for a little while. So if you have other questions that come up and you'd like to drop them in here, Rajan and Veronica are willing to answer those questions today. Or you could share them with us via email, and we will put together a nice Q&A to accompany the recording of this webinar and the additional information that will be posted at the ogallalawater.org website. So thank you again to our speakers and to everyone for joining, and we hope you have a wonderful afternoon.
Thank you.
Okay. Thank you.